So I just want to thank Dennis, too, for making this conference happen, and for Annette organizing it, putting up with my late submission of my slide deck and all of that. So also a big thanks to the people here at the house and for everyone who's been taking care of us. So it's an honor for me to be here, to get an opportunity to meet all of you this week and to hear all of your talks as well. So as Michael said, today I'm presenting a PhD proposal. So it's a bit of an outlier here. It hasn't started yet, scheduled to start at the beginning of next year. And we're still in the planning stages, but we're excited to present it like that and to hear feedback from you guys as well. So the team is myself and my two supervisors. Team is myself and my two supervisors from the University of Reading. That's Professor Julie Hawkins and Dr. Nandini Vasudevan. The project, as Michael mentioned, is also being developed with Dennis and Michael, and they've graciously offered their time to come on board as advisor and mentor and help me shape the project even to this point, so very appreciative for that too. We're also doing the project in collaboration with the UNOP Herbarium that you may have heard about this week down in Iquitos, where Juan Rez is the herbarium organizer there. So with all that said, again, genuinely feel really fortunate to be here and to present this at ESPD 55. I also want to mention my good friend and research partner, Derek Reffner, who's in Costa Rica. He's been a big inspiration for me over my life with teacher plants, and he's an amazing horticulturalist and really a source of knowledge for the project too. So although this project really is bigger than me, I want to share a little background on myself and my interest in the topic before I get into the rest of the components. So this is me in 1982, This is me in 82, just after my first experience with plant medicine. But for my mom's sake, I should say there's no psychedelics in that dropper there. But the drug I was given was called digoxin. So digoxin is a cardiac glycoside made from digitalis, also known as foxglove. I was given this for the first 2 years of my life for a pediatric heart condition. And it always stuck with me how it was instrumental to my survival, and how a plant could have such a profound effect on the human body. So it was much later that I discovered psychedelics and teacher plants and other fungi, and how those plants could have such a profound effect on the mind. So before getting into plants and studying plants, I worked in architecture and community planning. And I witnessed at that time just the stresses and mental health challenges that many of us are facing in cities today. And those experiences really shifted my outlook on the world, and I started wondering how I could contribute more to a world with less disease. So long story short, out of that passion for potential psychedelics and health and just the healing of humans, I decided I wanted to study plants full time, and so applied and got into this master's program at the University College of London. And that's the MSc in Medicinal Natural Products and Phytochemistry. Sadly, they have discontinued that. But some of you might know Michael Heinrich. He was the former convener of the program there. So at UCL, I wrote a dissertation on the antimicrobial activity of two entheogenic South American plants, Anadenanthera and Psychoactive viridis. And that was a lab study. So as I mentioned, I studied the chemistry of Psychoactive in the lab. And the experience of studying the plant, along with my own personal experiences with the plants, really got me curious about the molecules that were in them and what exactly the plants were all about. So, and at that time, I drank ayahuasca for the first time, and so really, especially got interested in what's in ayahuasca. And I remember having this thought that despite an amazing foundation of work from so many botanical explorers, scientists, and so many of you here today, there's still so much more to know about ayahuasca. And before the complexity of what we know scientifically about the plants matches the complexity of the ayahuasca experience. So I think we've all witnessed this global expansion that's been discussed here this week. We've all witnessed the global expansion of ayahuasca. And we've all heard about or witnessed the potential for it to be of great benefit to the collective human psyche, the human condition. However, I think it's a good time to take a pause and reflect and ask at what cost, if any, to the plants, to the communities, and to the cultures that surround them. So you don't have to look too far to see the topic being discussed in some of the popular literature. And with the rapid rise of consumption and then the looming pharmacology applications on the horizon, people are asking questions about the sustainable harvest, the plants, and in some cases even the composition of the brews themselves. So I'll just read a couple of quotes from one from ICEER's here, 2021. So this is entitled Plant Medicine Sourcing in Changing Times. And they address a few things, but here they say rising demands in Western countries is having an impact on accessibility for local use by indigenous communities who are witnessing the harvesting of plants at a higher rate than is ideal for healthy regeneration. Below here from Chacruna.com, the conservation of the diversity of vine types should be considered essential to maintain this important genetic resource. A couple others over there I won't get into, but just highlighting or questioning what's going on right now. So I want to point out that not every one of these articles is about harvest volume or scarcity. Important questions are also being asked about what species are being harvested and selected for cultivation or selected for the brews. So in our mind, these questions are equally as important to understanding the health of the plants, species, and potential biodiversity impacts. So I'll talk a little bit more about the scientific literature that we've started to get into as well in a future slide. So a step back here to briefly mention that this is not a new problem, or rather a new challenge for medicinal plants. So this data is from a couple of sources, the World Wildlife Fund and the International Union for Conservation of Nature. But they summarize that there are 50 to 80,000 flowering medicinal plant species worldwide, and 15,000 of those are threatened with extinction due to overharvesting and habitat destruction. So they do highlight what discussed earlier this week, climate change. They say, although this threat has been known for decades, the accelerated loss of species and habitat destruction worldwide, according to, from climate change is the implication, has increased the risk of extinction of medicinal plants. So that's on top of these numbers. The pressures are growing even greater. So getting into our proposed study, the question that frames the project here for me is what can be done to help preserve the cultural knowledge and biodiversity of ayahuasca? It starts with a study to document ayahuasca as it is today. And to do that by collecting a set of well-documented samples from a variety of sources for future characterization. And doing that with the aim to identify the plant components and phytochemical fingerprint of each sample, while also documenting a snapshot of the cultural practices surrounding their sourcing, harvest, and preparation. So these are the beginnings of research questions, I guess you could say, or hypotheses, but right now we're asking what plant species are in ayahuasca and how were they sourced and prepared, and what is the chemical result of the brewing of those plants. So I think we use the term ayahuasca in many situations now, but we want to ask with this project, what are we really talking about when we say ayahuasca, and what exactly is in it? So another way of thinking about this first question is also, what are the botanical variations that exist in the ayahuasca preparations? So we heard a really good talk from Geronimo and Andrea last night, but I think in research projects, ethics are almost always discussed at the end, but I think it's especially important for this kind of work to discuss it in the beginning and really consider it in the beginning of the project. So I'll present it today as a challenge that we're facing, a challenge that's really an opportunity. And that's, how do we design and conduct a rigorous scientific study of ayahuasca that honors the plants, the people, and the communities that have carried their knowledge. So as we form the study, we've assembled some principles to guide our process, and that's listed here: ongoing conversations with communities, prioritizing the preservation of plant medicines in the environments and communities where they originate, and that is from chacuna.com, if you know them, that organization. And also reciprocity through benefit and knowledge sharing of the data we're collecting. So to begin to discuss a little bit the beginnings of our literature review and the work that's out there that's been done on the topic, just to say that the knowledge base that we're drawing on is this, and it's looking into ethnobotanical explorations of ayahuasca and the plants, looking into the taxonomy of the various species, and then finally studies that use phytochemical analysis to investigate the brews and the plants themselves. So yeah, this is going to be a little small, but what's maybe even more important for this talk is from the scientific literature we've looked at. These are all studies, field studies of previous surveys, basically, of ayahuasca or yahe. So let's see. With these, like I said, I haven't fully reviewed the literature, but I think it's interesting to point out a couple things. One, I'll read a couple of these numbers. Most of the surveys have collected about 30 to 40 brews. Some plants, like this recent study, they collected 176 lianas. So that's just to kind of give you a landscape of what's happening. As far as we know, our study will be the first to use DNA meta-barcoding to look at ayahuasca plants. So we think that's exciting. And most of these studies that I was talking about that surveyed 30 to 40 brews were all from the syncretic churches. So I'll get into it later, but we want to source from a variety of different sources. And this is a quote that kind of summarizes or pushes us forward a little bit. But in spite of, this is from the most recent study of ayahuasca that did the 179 lianas and 38 brews. But they conclude, in spite of the globalization of ayahuasca use and the substantial number of studies showing a broad spectrum of therapeutic potential of ayahuasca, copy and beta carbolines, there are very few studies that examine the chemical profile of the beverage and plant obtained from different sources. So I'll get into the process and our methods here of the study. As I mentioned, our study is a survey to collect and document samples from the field. So the fieldwork location is Peru, and it'll be drawing on some of the relationships and groundwork that Michael has done around Pucallpa and Iquitos. So we would like to also collect samples from other locations to be able to learn more about the composition of ayahuasca and how it's changed as it's spread cross-culturally. Our goal is to collect a variety of different preparations, like I said, from traditional and neo-shamanic sources, with our target number being 100. So for samples for this study, we define them as source plants, brews, and we also want to collect some of the condensed brews, also known as bricks, that people are using. So we will collect the plants, ID them visually, prepare voucher specimens, and deposit those in the UNAP herbarium. We also would like to deposit living cuttings at the UNAP facility of some sort or another local site. So once we've documented the specimens, then we'll get into the analysis portion. And for us, for our study, that takes two forms. Like I mentioned, we want to do DNA metabarcoding to basically build lists of plant ingredients from the brews that we're sampling, as well as being able to use that in some capacity to identify the species of the plant specimens that we collect. Second, I know you guys have heard a lot about this week, but we will do a sort of classic phytochemical analysis, which involves using HPLC or LC mass spec to characterize the samples that we collect. So finally, over there towards the right, we are planning some pharmacology work at the University of Reading to investigate a few questions. One, if lab evidence can be established for some of the reported therapeutic effects of the brews, and if possibly any behavioral correlations can be seen between the brews prepared by different practitioners. So this could potentially imply unique applications for different brews in contemporary medicine. Also, we believe that some of our findings could be useful, yielding higher taxonomic resolution could be useful for biodiversity studies and understanding more about the potential threats that the plants face. So I'll go through this a little bit quickly, but this is just a distinction to say that we propose to use DNA metabarcoding, which is a different process than DNA barcoding. And this is a quote. This is actually a study that used, in a kind of similar way, used barcoding for a biodiversity study. But they describe DNA barcoding involves sequencing one well-curated individual at a time. Metabarcoding entails massive parallel sequencing of complex bulk samples for which morphological ID and curation is not practical. So this is from a commercial genomics company, but I think they describe it pretty well, and that's 4 simple steps to metabarcoding. But the first step is to isolate the DNA from different matrix. So in our case, our matrix is the ayahuasca brew. They prepare, to prepare the DNA, step 2 will be to prepare DNA metabarcoding libraries for the target taxonomic groups of plants. So we will have to construct our own library of reference plants at the outset by sequencing the plants that we collect or voucher specimens that we can find in herbarium. So once that's done, use a fancy benchtop equipment here like the MySeq to sequence, and then the raw data can be put into some pretty nice looking graphs here. And so I'll walk you through, this is a real world example from the literature of a study of herbal teas, but I think it's pretty comparable to what we want to do. So this top graphic here is from an Italian team at the University of Milan that used metabarcoding to identify lists of plant species in herbal tea samples. So they bought a bunch of herbal tea samples, brewed them up, and then did the DNA extraction. And from that extraction, they were able to then produce a list of plant species or essentially ingredients in the tea. So, and then the output of that, of course, was a list of species that they were able to publish. So again, the other arm of our study here is the chromatography or the phytochemistry. So briefly, just to say that HPLC, the output of that, or LC mass spec, is a chromatogram. So each of those peaks will represent a different compound that could be a compound in the plant or a compound we find in the brew. And this is from that big survey of ayahuasca that was just done, the 179 lianas. So they have pages and pages of their samples showing what alkaloids are in them and at what concentrations too. So I think it'd be really interesting personally to create a database, if you will, of these brew samples and the list of ingredients and associated alkaloids. And potentially to make correlations or to query the database, especially if we document local practices or local traditions around that. So all of this with the idea that it could help shed light on that kind of age-old questions about what's in ayahuasca, what are the admixture plants being used in ayahuasca. Or perhaps with the DNA work, it could even shed some light on the varieties of ayahuasca vine that we know indigenous people can identify, but that Western visual methods fall short on. So much like Laurel and Josep's project, it's very inspiring to hear what they're doing, but we want to ensure our data exists in a way that protects cultural information. And so we would use techniques like they used to anonymize using anonymous identifiers or not listing location data. So to summarize, I think it's fascinating to me how much more there is to learn about ayahuasca and the ayahuasca plants, and that someday that we could have botanical data that begins to match the complexity of an ayahuasca experience. So I want to emphasize that our goal is to learn more about, also with this project, to learn more about how we can serve as a platform to help with preserving biocultural knowledge and biodiversity. And we hope this study might be like a step in that direction and something that people could build on in the future, and one that's done respectfully in conversation with communities and also with the plants themselves. So thank you for listening.